information about the SBIR STTRs. SBIR stands for Small Business Innovation Research Award, and STTR stands for Small Business Technology Transfer Award. Both started very early in 1982 and 1992, so they're funding opportunities that have been around for a little while now. So if you see, this would be like the evolution of a specific research idea that you generate first in the lab with basic discovery research, that then you obtain specific data, that you have this breakthrough idea that you want to take further and commercialize it. That's when then we have like further steps into taking that idea into the commercialization steps. First, the proof of concept of that specific idea. Once that is obtained, we then have research and development for the development of a prototype. And once this is all resolved and clear, we can take it for scale-up manufacturing and sales and distribution. For the basic research discovery, we usually have research grants. They could be federal or private entities that give you or awards you funds to do that specific research. And then for the proof of concept and the research and development, the SBIR STTR programs exist. This is very important because in these two steps, there's high risk that it could work or it can't work. So why is this program vital for the development of that research idea? Well, because the federal government will be awarding you funds specifically to invest to take away the risk of the technology. You will generate more intellectual property of your technology. And also, these are sources of non-dilutive funding, which means basically it's capital to work on your technology without having to give away equity of or ownership of the company. Once these ideas are developed and you are on your way to commercialization, additional sources of funding can come into play, but these friends and family, founders, angel investors, venture capital, and strategic partners tend to be sources in which you have to give some of your equity in order to be able to receive that capital. So why is the SBIR and STTR funding mechanism great opportunity for both the university and the company? Because with this specific mechanism, you can propose a high-risk idea that has never been proven before for a specific unmet need. This is a technically difficult project that requires expertise, both from the business perspective, which in my case would be Puerto Rico, and from the research perspective, which in my case would be us from UCC, my team at UCC. Big companies do not embark in these high-risk ideas because they have a more complex structure, and also they prefer that small companies, such as Puerto Rico, do this type of research and that it is financed through federal funds. And why? Because if the government takes the risks and decides to invest in you, then it means that you're doing something right and it is worth to take this idea to the market. This idea will be used, or these funds will be used for research, not a marketing strategy. And there are two phases. Basically, you define the research strategy that describes what will you do to take your idea further and do that proof of concept in phase 1, and then develop a prototype in phase 2. I should say that the Puerto Rico Trust has a specific office designated for the development of this type of funding, and you will hear a little bit more about it in a little while. So let's talk about Puerto Rico. So what you see in the background here is Ganoderma. Ganoderma lucidum is a medicinal mushroom that has lots of properties. We at Universidad del Caribe have studied it for more than 10 years, and we have developed evidence-based research specifically on anti-cancer properties of Ganoderma. The local company, Puerto Rico, is an agricultural company that is basically a company that cultivates mushrooms, but they're edible mushrooms, the ones that you cook in your kitchen. However, they participated in Pre-18 from Parallel 18, which is also from the Puerto Rico Trust, and decided to embark into a research and development venture. They seeked advice at the Puerto Rico Trust, and they referred them to me. So I'm at UCC, and at UCC I work with specifically Ganoderma lucidum. I have worked at UCC since 2007. I've submitted many grants, and also have published many papers specifically on the properties of Ganoderma. So I've basically done research on evidence-based effects that this mushroom has in aggressive breast cancers, including inflammatory breast cancer and triple-negative breast cancer. We have shown how Ganoderma inhibits selectively cancer cell viability, not only in cells but also in mice, and it also affects stem cell motility, invasion, and metastasis. We also characterized Ganoderma and published this in 2019 and submitted a patent, which is now published, on the effects of specific compounds derived from Ganoderma that also have anti-cancer properties. And I also have papers, additional papers, that are also detailing characterization of populations here in Puerto Rico for specific catastrophic diseases such as cancer. So it was basically a very good pairing because of the intention of research and development of Ganoderma. So the initial meetings between Puerto Rico and myself led to a memorandum of understanding. And this is something that everybody that decides to embark in research should have. Independently, if it's a company or if it's a colleague, you should have this in place because these are the rules of the game. What's going to happen in which place that you're doing the research. Also, we had frequent meetings to discuss which agency and funding opportunity we were going to apply for, and the generation of preliminary data for that specific grant, as well as to write the sections of the grant application. So what did we do? We first applied for the Puerto Rico Trust, an advanced research grant, and we did not get funded. But we did get great reviews, and with those reviews, what did we do? We submitted a pre-application to the NSF. The NSF told us, okay, you can submit a full application, but at the same time, we also submitted the USDA SBIR in a somewhat different proposal, and we were awarded. And therefore, we decided to go ahead with the USDA award. So this is basically what we're proposing to do. So Puerto Rico is going to be in charge of the cultivation. Then of the cultivation of the Ganodermas. And I'm going to explain to you a little bit more about those specific Ganodermas in just a little bit. LSU, which is my partner institution, is going to do the extraction of the bioactive compounds, and then we at UCC will test them. So first of all, Puerto Rico, it's going to do the cultivation process as well as the production process. They have different species that are neotropical species of Ganoderma. They include Ganoderma martinicense, Ganoderma multiplicatum. We have up to 6 native species. Why? Because the neotropical species, because they're in a different environment than those that are in China, for example, are very different. The environment, the temperature, the substrate, the humidity, everything changes the properties of these specific mushrooms. So the idea is to cultivate them and then extract the bioactive compounds and develop a prototype for different diseases. So Sebastián Sarardía is the founder of Puerto Rico, and Kurt Miller is the field specialist. He's the one that collects these Ganodermas in the field in different parts of Puerto Rico. So then, with the help of the Puerto Rico Trust Biodiversity and Tropical Division, we did the first extractions of Ganoderma, and then they basically referred us and said, so you need a specific medicinal chemist to be able to do the extractions and to characterize further these species. So we partnered with Fatima Rivas at Louisiana State University, and she basically is doing bio-guided fractionation of all of the different Ganodermas in order to then obtain specific bioactive compounds. Then those bioactive compounds are shipped, well actually, the fractions are sent to me, and I test them in the lab. So here's some data that we used for the preliminary data for the grant, actually. So these graphs, on the x-axis is the concentration of the mushroom, and it's all in milligrams. And in the y-axis is the cell viability of specific cancer cell lines. This is Ganoderma martinicense, Ganoderma multiplicatum, both neotropical species, and this is Ganoderma lucidum from a commercially available source. As you can see, the viability is drastically reduced with the neotropical species extracts, but it isn't the same as the Ganoderma lucidum. So they are much more effective in cancer cells than the commercially available source. Furthermore, we then tested them in non-cancerous cells, which is basically what we find with our other studies. So in non-cancerous cells, we see that the cells do not die, but with the cancer cells, they do die as you increase concentration. So this is selectivity, and this is a very, very good characteristic from the mushroom that permits us then to create additional prototypes. There is therapeutic window. So what are the challenges and benefits? Challenges, the pandemic hit. So I had to close my lab. They had to stop working in the agricultural venture. Another challenge is that it's a young company. They had to do a lot of things for the first time, and that takes time to be able then to submit the proposals, etc. And the preliminary data was a little bit challenging to obtain it because it was right in the middle of COVID. They had a 400 square feet house. They produced 100 pounds of mushrooms weekly, and I had to close the lab because of COVID. So it was limited testing. But that also gave us opportunity. It gave us time to write, because we needed to write those grants. It gave Puerto Rico the availability of doing pivoting, changing directions. Where are they going? To do additional research ventures. The research and production, now they have specialized areas because they have a huge specific space of 4,000 square feet. They have areas for fructification, incubation. Now they produce 0.5 ton of mushrooms weekly. And I was able then to increase the research workforce by hiring other people, publishing, and submitting grants. So we were able to de-risk the technology. That increases the value of the technology. And also we are submitting additional disclosures. What does that mean? We're submitting additional data to increase also what we know about these species. We are generating ideas to create new SBIR grants, and also ideas to do research and development of other products. Puerto Rico has their own ideas to create other research ventures, and also I'm glad to say that we at UCC I am creating a company to then commercialize what we found with the bioactive compounds from the other project of Ganoderma. So I could not do anything without my team. These are all undergraduate students that work with me: Alia, Adriana, and Paola, my postdoc, Luz Arroyo. These are all the people who have been involved in the Ganoderma projects. These are my collaborators. This is the funding sources. Thank you so much.